Namaste, this is JVN Dr. Vishal Saxena, Associate Professor in Mathematics and Statistics in Jayoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss on the module chi-square test of goodness of fit test and homogeneity of samples. As all we know that chi-square test is a very important test and similar to like the other hypothesis test, statistical test, t-distribution or the ANOVA and the other distribution tests like the z-distribution test. This chi-distribution test is an important part of biostatistics because in the biostatistics the various kind of results are to be tested and for this just we are required to test these results with our some statistical techniques, statistical tests, and the chi-square is one of that test. So chi-square chi-square test determines whether there is a significant difference between the expected and the observed frequency of one or more categories for the contingency table, and this test can be divided into three parts. The chi-square test of goodness of fit means the goodness of fit test. Second test for the chi-square test is the chi-square test for independence, and the third test of chi-square is the chi-square test of homogeneity. So in the today's session we are discussing about the two tests of chi-square test of goodness of fit test and the chi-square test of homogeneity. These two are very important tests, and in the first of the test, the chi-square goodness of fit test, it enables us to compare the observed sample distribution with the expected probability distribution, means how the theoretical probability distribution like a binomial or the Poisson and the normal distribution test distribution is significantly different or fit for the empirical distribution, while the second test which we are discussing about today, that is the homogeneity test. That is the test which explain the significance of difference, the significant difference of frequency counts for the different populations. That means it will determine whether the frequency counts are uniformly distributed over the different populations or not. So for the today's test we are going to share some like mathematical concepts with examples and mathematical terminologies, and for this just we are going to share some slides today. Initially the chi-square goodness of fit test is mentioned there, just we have explained it with the definition. Initially this test chi-square goodness of fit test determines how well the theoretical distribution such as the normal, binomial or the Poisson test distributions fits the empirical distribution. So that is the main objective of this chi-square goodness of fit test, to apply for our results, our obtained data, that the observed sample distribution with the expected probability distribution, that these are significantly different or not. And for the chi-square goodness of fit test, initially we have to assume a null hypothesis in which just we will assume that that there is no significant difference between the observed and the expected value. Means just we will assume that there is no difference between the observed and the expected value, while in the alternative hypothesis we will assume that there is a significant difference between them. Now in the chi-square goodness of fit test, we will take a chi-square statistic, and it is given by summation O minus E whole square upon E, that is the chi-square statistic. And according to the degree of freedom, the chi-square distribution can be taken, like we are given that a table binomial distribution for the Poisson and for the normal distribution with the number of constraints 1, 2, 3 and degree of freedom. Now in the part of the hypothesis testing, it is just similar to the other tests like the T distribution and the ANOVA, and in which we have to find the chi-square value, and we have to check for the particular level of significance that whether this observed value will be greater than or greater than the expected value. If it is greater than the expected value, then definitely the null hypothesis will be rejected, and if the observed value is less than the expected value, then it will be accepted if the observed value is less than the expected value. So these are the cases for the hypothesis testing, and by this we can calculate our results. Now next is the chi-square test of homogeneity. This is a test, and in this test there is a single categorical variable. Just we are having a single categorical variable and taken from the two or more different populations, and this test can be used to determine whether frequencies counts are distributed identically or not across the different populations. So that is the main objective to apply this chi-square test of homogeneity, and that whether the frequency counts are distributed uniformly or not across the different populations. For example, in a survey of TV viewing preferences, we might ask respondents to identify their favorite program, and this question we might ask the same question of two different populations, means males and females, and we could use a chi-square test for the homogeneity to determine whether the male viewing preferences differ significantly from the female viewing preferences. So just it means we are having a one categorical variable, that is the viewing preferences, and the two kind of populations are there, means males and females, and that's why just we can apply the chi-square test of homogeneity for this example. And when to use chi-square test of homogeneity? For each population, the sampling method is a simple random sampling, and the variable under study is the categorical. So the first condition is that the variable should be categorical, and the second condition is that the sampling distribution, the sampling method must be simple random sampling. No other kind of sampling should be taken. If sample data are displayed in a contingency table, the expected frequency count for each cell of the table is means at least 5, means minimum 5 for the each cell entry for the contingency table. So these these are the conditions for the application of homogeneity test, and in this test how to apply the process. So just we will find here every hypothesis test requires that analyst to state in a null hypothesis and alternative hypothesis. The hypotheses are stated in such a way that they are usually mutually exclusive, and initially that means we have to find we have to make the hypothesis, the null hypothesis and the alternative hypothesis, and just these two must be mutually exclusive. So that is our the starting point. And at the specified level of categorical variables, the null hypothesis states that each population has the same proportion of observations. So that should be our null hypothesis. That means each population should have the same proportion of observations, and the alternative hypothesis is that at least one of the null hypothesis statement is false. Then formulate an analysis plan. So just we have to apply our plan of analysis for this test, and for this we will take our level of significance, that means 0.01 or the 0.05, 0.10. Test method: use the chi-square test of homogeneity to determine whether observed sample frequency differs significantly different from the expected frequencies specified in null hypothesis. That is always a part of our analysis. Just we have to again use the chi-square test method for homogeneity for seeing whether the observed frequencies are different from the expected frequencies or not. Then we have to find our degree of freedom. Degree of freedom is given by r minus 1 into c minus 1. R means the number of populations and c represents the number of labels of categorical variable. And then after this, that is very important, how to find the expected frequency. The expected frequency is given by ER comma C. The ER comma C is given by NR into NC upon N. NR into NC upon N. And where, what are the notations there? ERC is the expected frequency count for population for population R at level C of categorical variable. NR is the total number of observations from population R, and NC is the total number of observations at level C. So now the finding of the test statistic. The test statistic is given by chi-square, and chi-square is equal to ORC minus ERC whole square upon ERC. That is the formula for the test statistic. And after this the p-value will be calculated. The p-value is the probability of observing a sample statistic as extreme as the test statistic. And since the test statistic is a chi-square, use the chi-square distribution calculator to assess the probability associated with the test statistic. And finally we have to interpret our result with the observed value and with the p-value. So if the observed value is greater than the p-value, then our hypothesis, null hypothesis, will be rejected, otherwise it will be accepted. So that is the involves comparing the p-value to the significance level and rejecting the null hypothesis when the p-value is less than the significance level. So that is the condition. And so in the today's session we have discussed about the two tests of the chi-square, means chi-square, the goodness of fit test, and the chi-square homogeneity test. These are very important tests, and students are advised to take problems, numericals and objective questions of these topics to just for the preparation of your exams and competitive examinations also. I am concluding my today's session with this. Thank you.